Hi, I'm Jake. Thanks for joining. And thanks to Yamina and Dr. GPCR for this opportunity to present today. We'll cover fluorescent biosensor assays that allow you to record GPCR signaling in living cells. Being able to capture GPCR responses in real time means that you can use kinetic analysis to get more pharmacological insight. These biosensors give the ability to capture multiple time points in a single well and easily compare different ligands. Montana Molecular has developed genetically encoded fluorescent biosensors for GS, GI, and GQ pathways. These biosensors measure changes in cyclic AMP, PIP2, diacylglycerol, calcium, and beta-arrestin and can be expressed in a variety of cell types. Our sensors are compatible with fluorescent imaging systems or automated plate readers, and the rest of the data in this presentation are from fluorescence plate readers, but these movies illustrate the spatial and temporal dynamics of biosensors in living cells. The signal-to-noise ratio from these sensors is very high, resulting in high Z' values. Because each sensor uses a single fluorescent protein, the sensors can be combined in the same cell to measure multiple signals simultaneously. The diacylglycerol sensor is built on a PKC delta backbone and changes in response to activation of the GQ pathway. Bright red and green fluorescent versions are available, and there's a list of recent publications using this sensor available on our website. The CADIS assays detect GS and GI mediated changes in cyclic AMP. This sensor is built on an EPAC backbone with a single circularly permuted fluorescent protein. CADIS sensors that are targeted to primary cilium and other microdomains, as well as Cree-responsive versions, are also available. Here too, a list of publications using both red and green versions of CADIS can be found on our website. CADIS assays for cyclic AMP make great tools because of the high temporal resolution and high signal-to-noise. This data is collected in collaboration with Patsy McDonald's lab at the Moffitt Cancer Center on a Biotech Synergy MX fluorescence plate reader. Our beta-arrestin sensor shows a decrease in fluorescence to indicate arrestin activity and has been optimized for use with several different GPCRs. Here we're looking at a series of bias ligands for the GLP-1 receptor. The arrestin sensor can be used to effectively measure both partial and bias agonism. The analytic methods developed by Sam Hoare at Pharmacanics are ideal for measuring partial and bias agonism, so be sure to catch Sam's presentation, Analyzing Kinetic Signaling Data for GPCRs. Montana Molecular sensors and many GPCRs are packaged in BACMAM. If you've never heard of BACMAM, it's worth knowing about this system. BACMAM is a BSL-1 viral vector that allows efficient delivery to practically any cell type, including primary cultures. It has a large carrying capacity and lets you control the level of expression of a receptor or a biosensor, which helps optimize your assay. The protocol is simple and there's minimal liquid handling. Here's a few reasons to be happy. Now you can record GPCR signaling for kinetic analysis in the living cells of your choice. Montana Molecular produces a variety of fluorescent biosensors for GPCR discovery, COVID-19 research, and other applications, as well as assay development and optimization services. Thank you so much for your time. You'll find more info about these biosensors for GPCR signaling on our website, montanamolecular.com, and we hope that we'll hear from all of you. Thanks again.